So the next question is question number 46, and it says that the embryological support for evolution was disapproved by. So this is a kind of a direct question that you can say, and this embryological support was actually disapproved by a scientist, and that scientist is Carl Ernst von Baer. He is the scientist who actually disapproved this concept of embryological support, and he said and noted that the embryos actually never pass through the adult stages of the other animals during the process of embryonic development. So this is a kind of a direct question, as you can say. So we can say that embryological support for evolution, just a moment, for evolution was disapproved by von Baer, actually. Von Baer, and he noted, or he actually stated, that the embryos, they never pass on. That embryos never pass through the adult stages. So through the adult stages of other animals during the process of embryonic development. So during embryonic development. All right, so this was one of the earliest of the theories that you can say that has been given. However, this wasn't supported by von Baer. All right, so that is the case. So these scientists, the rest of these scientists, they have different contributions. Like Oparin has been related with the chemical theory of evolution. Charles Darwin is related with, you know, your theory of natural selection. So other scientists have other contribution, but it was the von Baer who didn't supported this theory of embryological evidence. All right, so let's move on to the next question. Then bilaterally symmetrical and acoelomate animals. Now this is a question that comes from the animal kingdom, as you can see. So it's talking about here the organisms which are bilaterally symmetrical, and apart from that they are acoelomate also. All right. So if we just look into the various, you know, phylums of the animal kingdom, then the bilaterally symmetrical, and that's why they are acoelomate. That means they are not having any coelom. All right. So out of all the options that has been given over here, that means Platyhelminthes, Esselminthes, Annelida, and Ctenophora. All right. So if we look into the options, the correct option or the correct answer is exactly the Platyhelminthes, because Platyhelminthes, they are bilaterally symmetrical. They are having coelom and actually having organ system level of organization along with the triploblastic organization, you can say. All right, so Platyhelminthes is the correct answer, because Platyhelminthes, so these organisms, they are bilaterally symmetrical, All right. That means the body gets divided into equal parts passing through a central plane only. So bilaterally symmetrical, then they are triploblastic. Triploblastic means that they are having all the three germ layers, ectoderm, mesoderm, and endoderm. So that's why triploblastic. Then they are acoelomate also. That means, just like acoelomate means that they do not have any kind of a body cavity, which is coelom. All right. So that's why they are acoelomate animals. All right. Now, if you just look into the other categories, if I tell you, so SL minthus is one such category that is bilaterally symmetrical, but that is actually pseudocoelom. So if you just look into SL minthus, so SL minthus they are bilaterally symmetrical, but they are pseudocoelomate. They are also called as nemathelminthes or ringworms. Pseudocoelom. Pseudocoelomate means they are having a false coelom or a body cavity. That means the mesoderm, the entire mesoderm is not present, but mesoderm is actually present in the form of patches called mesoglia between ectoderm and endoderm. So they are pseudocoelomate. Then regarding Annelida, if we see, then Annelida also, this is bilaterally symmetrical. This is bilaterally symmetrical, and apart from that it is coelomate. So this is the case. And the last option, so this is again not the option, this is not the option, and the last option turns out to be the Ctenophora. So if you just look into Ctenophora, so the members of Ctenophora that comes just above the platyhelminthes, just before the platyhelminthes. So Ctenophora are actually the organisms which are radially symmetrical. So these are radially symmetrical and acoelomate. All right. So this is the correct reason. So again, this is not an option. So option A is the correct answer. That includes all the tapeworm organism. That means platyhelminthes. All right. Now moving ahead. Question number 48. So question number 48 says that which of the following would help in the prevention of diuresis? All right. So which of the following action out of all the four action is going to prevent the process of diuresis? So basically, if you just look into the options, the first option says that resorption of sodium and water from the renal tubules due to the aldosterone, if you're going to see. Then there is atrial natriuretic factor, ANF, causes vasoconstriction. Then decrease in the secretion of renin by the JG cells and more reabsorption of water due to under-secretion of ADH. Now, out of all the options, it is saying that which process is going to prevent diuresis. All right. So diuresis is basically going to be prevented. All right. It is because of the resorption of water and sodium ions from the renal tubules of the aldosterone. This is because adrenal cortex, it secretes the mineralocorticoid. All right. And the mineralocorticoid is the aldosterone over here, which is ultimately going to increase the process of resorption of sodium ion as well as water. So that's why the correct reason will be that adrenal cortex that secretes mineralocorticoid. And that mineralocorticoid is the aldosterone. That is aldosterone. All right. And this aldosterone only is responsible for increasing the resorption of sodium ions and water, which you can say increases the reabsorption of sodium ions and water through the kidney tubules. That means the renal tubules. So from renal tubules, thereby preventing diuresis. So thereby preventing diuresis. All right. So this is the case. So out of all the options that you can see, the correct answer is option number A, which is resorption of sodium ion and water from the renal tubules due to aldosterone. All right. Now, let's move on to the next question, which is question number 49. So question number 49 says that you have to, again, this is a match the column kind of a question that has been given. So you have to match the following columns and select the correct option. All right. So there are some, bioactive molecules that are released by different kind of microbes has been given. So column 1 is actually, as you can see, the A, B and C, D option. So these are actually the microorganisms, all right, different origin. And column 2 basically indicates the different bioactive molecules. So these all are actually the bioactive molecules. If you remember, then these are all bioactive molecules that is released by different microorganisms. All right, now let us take a look at what is the criterion and what is the source. So this is a kind of a direct question that doesn't requires any kind of explanation. So as you can see, the first category is a bacterium, which is Clostridium butylicum. All right, so this is Clostridium butylicum. Clostridium butylicum is a source of butyric acid. So as the word is itself suggesting, it is butylicum. So this will be the source of butyric acid. So here will be the option A. All right, so this will be the source of butyric acid. Then Trichoderma polysporum. All right, Trichoderma polysporum is a source of cyclosporin A. That means this is a kind of a fungus, and this category is a source of cyclosporin A. All right, this is the case. Then Monascus purpureus. Monascus purpureus is actually, again, this is a category of a fungus, and this acts as a blood cholesterol lowering agent over here. That means this is the option of C. And the last turns out to be a fungal category, which is Aspergillus niger, and this is a source of citric acid. All right, so citric acid. So if you just look into the various kinds of options, so let us just take a look again. So we have seen that out of all the options, so A will be belonging to the butyric acid over here, Clostridium butylicum. That means A will be in correlation with 2. Then B will be in correlation with 1. That means this one. So cyclosporin A, as you can see, Trichoderma polysporum produces cyclosporin A. So this is actually a immunosuppressive agent. All right, during the organ transplant patient, this cyclosporin A. All right. Then C goes with 4. As you can see, Monascus purpureus, this is going to go with 4, this one, and D will go with 3. So our correct option turns out to be option number A. All right. So this is a kind of a direct question, which you can see that actually doesn't require any explanation. So we have done it like here only. All right. So that is the case. Now, let's move on to the next question, which is question number 50. Now the question number 50 says that some dividing cells exit the cell cycle. Some of the dividing cells, they are going to exit the cell cycle and they actually enter into a vegetative inactive stage. And that inactive stage is called as the quiescent stage. So this process occurs at the end of. All right. So basically, if I tell you, then, the cells that actually, doesn't enters into the division phase and it exits the cell cycle, it basically, those cells are very specialized. Those cells can either be the heart cells and neuron cells that doesn't goes, into a division phase, that doesn't enters, in fact, the cell cycle also. So for that reason, if you just see, then as soon as these dividing cells, this process, that means this inactivity process, you can say, or this exit process, it actually occurs at the end of G1 phase. This usually occurs at the end of G1 phase, or you can say it is basically in between these two processes. That means G1 phase and M phase. This is in between these two processes because it is the end of the M phase and it is the beginning of the G1 phase. All right. So for that reason only they are going to exit the cycle. So indirectly we can say, but it is more since it is a kind of a dilemmatic answer. Both of the options are available. So we will actually go with this G1 phase. All right. Because if you just have the sequence, then G1, then there is S, then G2, and then there is M phase. All right. And out of these phases, so we know that this belongs to the interphase stage. This is the interphase stage, and this is the cell division phase. This is a cell division phase. So if a cell has to exit the cell cycle, then it will exit it either at this stage or at this stage. All right. So at the end of the metaphase, you can say, or at the beginning of G1 phase. So if it has to enter it. So if we are going to correctly like define it, so it is going to be the G1 phase. All right. So the correct answer will be G1 phase. So you can say that this process occurs at the end of M phase and beginning of G1 phase. That's why. All right. So answer will be, correct answer will be G1 phase. Clear? Now, next question. So question number 51 says that in which of the following techniques the embryos are transferred to assist those females who cannot conceive. Remember, it is here talking about the embryos. There is the transfer of embryos. So that means already the fertilized products, the fertilization has already been taken place in the artificial environment. That means in the lab conditions. So that's why this technique basically is going to involve some advanced techniques of ART. All right. So if you just look into, so the processes that has been enlisted over here. So the first process tells us about the gift and the zift, if you can see, basically. So all these are the techniques of ART. We know that all these are the techniques of ART, which are assisted reproductive technologies, you can say. Assisted reproductive technologies, which is going to assist a couple in conceiving or having a child. Technologies. Clear? Now, if you just look into the first option, which says gift and zift. All right. So this gift stands for gamete intra-fallopian transfer. That means the gamete will be transferred, ovum will be transferred to the female who actually cannot conceive. But here it is talking about embryos. It is not talking about the gamete. That means the ovum. So again, this is not an option. All right. And what does ZIFT stands for? ZIFT stands for zygote intra-fallopian transfer. That means the direct part of the fertilization, the direct result of fertilization that has to be transferred, which is your zygote. Again, this is also, if we can say, so this ZIFT, that means zygote intrafallopian transfer, this can be an option. Why? Because this zygote only it will depend that whether the early embryos are being transferred to the fallopian tube, or the late embryos are being transferred. Because in categories of ZIFT, if we say, then there are two categories: either the early embryos, that means the morula stage, or the later embryos, that means after the morula stage. All right, so this can be an option, but since GIFT is not an option, so that's why it is cancelled out. Then the second option is called as ICSI and ZIFT. So again, ZIFT is an option, but what does ICSI stands for? ICSI stands for intracytoplasmic sperm injection. Again, this is also not an option, because here the sperms will be injected into the cytoplasm of ovum. So again, this is not an option. So this is also rejected. Then the third includes the category of GIFT and ICSI. So as already stated above, both of them again are not an option. So this also won't be an option over here. This also won't be an option, and this won't be an option. Clear? Now the last turns out to be ZIFT and IUT. All right. Now this IUT, this stands for intrauterine transfer. And anything or any fertilized product which is directly transferred to the uterus, that has to be little advanced. And that advanced stage is only in case of the intrauterine transfer, the embryos are transferred only. Embryos are transferred over here. All right. And in case of ZIFT, we have already seen that this can also be an option of embryos transference. All right. So that's why the correct option will be ZIFT and IUT. All right. So this is the correct, reason. Now, next question. Now, in the next question, it says that which of the following statements are true for the phylum Chordata? Now, you have to enlist the correct statement regarding the following phylum Chordata. So in the first statement, it says that in Urochordata, the notochord extends from head to tail and is present throughout the entire life. All right. So it says that in Urochordata, it actually, just a second. Yeah. So in Urochordata, the notochord is extending from head to tail while it is present throughout the entire life. All right. So this is the first statement. Second statement says that in vertebrata, the notochord is present during the embryonic development. This is actually a correct statement, you can say, because notochord in vertebrata, the notochord is present during the embryonic period, and which later on actually converts into vertebral column. This notochord will develop into vertebral column during the adult. All right. So that is the case. So this is a correct answer, statement, you can say, but this is not a correct statement, you can say. All right. Now, the next statement says that in case of the, chordates, you can say, the central nervous system is dorsal and hollow. Again, this is also a correct statement. So in case of chordates, the central nervous system is considered to be dorsal as well as in hollow. All right, so this is also correct. Now, chordata is actually divided into three subphyla, hemichordata, tunicata, and cephalochordata. Again, this is a wrong statement. All right. This is again a wrong statement. All right. So basically, if you're going to see the categorization, so chordata is primarily divided into two different categories, cephalochordates and eurychordates. So there are only two categories in which it is divided. All right, so this is a wrong statement. So out of all the cases, if you will see, then if we just, the option B, if you see, then invertebrata, notochord is replaced, is present during embryonic development, which is later on replaced by vertebral column, and that to where? In adults, you can say, in adults. All right. And regarding the C option, so C option is also correct, that in chordates, the central nervous system is dorsal and hollow. So this is it. All right. So this is the reason. So out of all the options that you can see, option C is the correct answer. That means B and C. Clear? So this is the case. Now, moving ahead to the next question. So next question says that snow blindness in Antarctic region is caused due to. All right. So the options given, this is a question from your environmental awareness chapter, standard 12. So as you can see that it states that snow blindness in the Antarctica region is caused due to. This is because of the inflammation of cornea due to heavy dose or high dose of UVB radiation, then high reflection of light from the snow, damage to retina caused by infrared rays, and freezing of fluids in the eye by the low temperature. So out of all the option, again, this is a very direct question that has been strictly, directly given from your NCERT. And it has been written that snow blindness is actually a disease that is related to cornea. All right. And this cornea is primarily affected because of heavy dose of the UVB radiations over here. All right. So cornea, what does not in human eye, the cornea is actually responsible for absorbing the UVB radiations and thereby resulting into inflammation of cornea that results into snow blindness or cataract or any kind of eye disease. So the correct answer will be this one. That means option number A. So what you will write is that, so what's the correct explanation is that UVB radiations damages the DNA, resulting into mutation. Resulting into mutations. Now, what does it do in case of human eye? So in human eye, if you're going to see, so in human eye, cornea absorbs UVB radiations, and so it's high doses. And so its high dose causes inflammation, or you can say swelling, inflammation of cornea, leading to snow blindness or cataract, etc. So this is the correct reason. It is because of the UVB radiations only that results into damage in the cornea or inflammation in the cornea, causing snow blindness. All right. Now, next question. Now, again, this is a match the column kind of a question that has been given. All right. And you have to identify the correct option. All right. Now, so let us just match it up with the, correct option. So the first column, as you can see, the first column is correlatable to the different categories of glands that has been given. All right. And the column 2 is related to the, diseases, various kinds of diseases that is either caused due to deficiency or caused due to excess secretion or hyper secretion of that particular hormone. All right. So let us start with the very first disease, which is the very first gland, which is going to be the pituitary gland. So if you just look into pituitary gland, so pituitary gland is actually, related to, yeah. So in case of your pituitary gland, if you're going to see, just a second, yeah. Now, the column 1 and 2 has been given. Let us take a look that what are the deficiencies or hypersecretion results into. All right, in the body. So if you just look into the very first category, which is the pituitary gland. All right. So basically, if you just look into the disease that is relatable to pituitary gland, so diabetes, out of all these disease, diabetes insipidus is because of the hypo release of the hormone that is called as the anti-diuretic hormone. So you can say that diabetes insipidus is caused due to hyposecretion. Hypo means under secretion. Hyposecretion of a hormone which is called anti-diuretic hormone. From the posterior pituitary. All right. So this option is correlatable to pituitary gland. That means insipidus is correlatable to pituitary gland. All right. Now, the next is the thyroid gland. Now, in relation to the thyroid gland, if you're going to see, then the thyroid hormones are usually T3 and T4 hormone, which is called as triiodothyronine and tetraiodothyronine. And it is because of the disease which is caused because of the excess secretion of thyroid gland is the Graves' disease over here. All right. So Graves' disease is caused due to excess secretion of thyroid hormones T3 and T4. All right, this is the case. Now, option C, which is the adrenal gland. So adrenal gland is actually correlatable to, you can say, Addison's disease, and because this Addison's disease is caused because of the hyposecretion of the hormone from the adrenal cortex. All right, so Addison's disease, you can say Addison's disease is caused due to hyposecretion of hormone from adrenal cortex. All right, that is the case, which they usually release corticoid hormones. So that is the case, and the last will be your pancreas. So pancreas is basically related to diabetes mellitus, and this is again the hyposecretion symptom that has been seen. So this diabetes mellitus is because of the hyposecretion of insulin from the beta cells of the pancreas. All right, so the last will be diabetes mellitus. Now this is due to hyposecretion of insulin from beta cells of pancreas. All right. So now, if you just look into the options, matching options, so A will go for third option. A will go for third option. Then B will go with first option. Then C will go with fourth option, and D will go with second option. So our correct answer is 2. That means B here. All right. So this will be the case. Now, the next question is that which of the following statements is correct? Now you have to mark out the correct statement. So adenine pairs with thymine through one hydrogen bond. Adenine pairs with thymine through one hydrogen bonds. Then adenine does not pair with thymine, and adenine pairs with thymine through two hydrogen bonds. Now, let me tell you, according to the ATGC rule, so there are 4 nitrogenous bases which are found in DNA. So 4 nitrogenous bases found in DNA. Clear? Now, those 4 nitrogenous bases are adenine, then guanine, thymine, and cytosine. All right. Now these two, they are called as purines, and these two are called pyrimidines. Now how the base pairing is done? So the base pairing is done between a purine and a pyrimidine. All right. So if you just look into the base pairing, so adenine will be forming 2 hydrogen bonds with thymine, and guanine will be forming 3 hydrogen bonds with cytosine. So this is 2 hydrogen bonds, remember. So out of all the options that you're going to see, option D is the correct answer. All right. And all these statements are wrong. Clear? So this is the case. Now, next question. Question number 56. Now you have to match the column with the essential elements, concerning essential elements with that of their functions in the plan. All right, again, this is a kind of a direct question that has been given. All right, so iron is basically, you can say, is one such essential, element, micro element, you can say, that is primarily responsible for that is required for the chlorophyll biosynthesis. All right, so iron is very important for chlorophyll synthesis. So iron is important for chlorophyll synthesis. All right. Then the next is B, that is zinc. Zinc is one such, micro element or a micronutrient, you can say, that plays a very important role in indole acetic acid, which is a form of oxen. This is actually a form of oxen that is required in its synthesis. So here it will be option number B. So zinc is actually needed for oxen synthesis. Oxen synthesis. All right. Then option C says boron. Boron is again a micro element which is required in pollen germination. They have a very important role in pollen germination. All right, so here will be the option C. So boron is related to pollen germination. That actually helps in establishing pollen pistil interaction, because it's a kind of a chemical, you can say. So for pollen germination. Then the last option that turns out to be the manganese, and manganese, we know that manganese play a very important role in the photolysis of water, because manganese along with calcium ions as well. So they play a very important role in photolysis of water, thereby liberating oxygen. All right, in the process of light reaction of photosynthesis. So manganese is related to photolysis of water. All right, so let's now match up all the options. So A is going to go with third option. A will be going with third option. B will be going with fourth option. Then C will be going with second option. And D will be going with first option. So our answer will be option number B over here. Clear? Now, next is, the next question is that which of the following is not an inhibitory substance governing the seed dormancy? All right. So these given below are some of the, you can say plant growth regulators that has been mentioned, and you have to, explain out the inhibitory substance. That means not an inhibitory substance means that this is going to actually promote the process of seed germination. All right, so this will actually break the seed dormancy, you can say, in other words. So options given are abscisic acid, phenolic acid, para-ascorbic acid, and gibberellic acid. All right, now out of all the options that has been given, so if you just remember, then gibberellic acid is one such, that means GAs are one such hormone that is responsible for breaking the seed dormancy and will promote the germination of seed. All right, by mobilizing or by activating the synthesis of alpha amylase enzyme in the seed, which is going to, which is actually going to mobilize the starch molecule into simple sugar. All right, so that's why the correct answer here will be that gibberellic acid, that means GAs, break seed dormancy. So since this is going to break seed dormancy, thereby activating the synthesis of alpha amylase enzyme, which now this alpha amylase enzyme, let me tell you that this is only released in case of, this is found actually in case of monocots. If you're going to see now, then this enzyme is being released in the aileron cells of the monocot, seed. So which ultimately breaks down starch into simple sugar, and those simple sugars will be ultimately absorbed by seeds and will ultimately undergo the process of germination. So that's why our correct answer turns out to be this, because all these substances where you can see over here now, these are actually the substances or the PGRs which is going to promote the process of seed dormancy, because GA and ABA, they are antagonistic to one another. This we have seen. Since GA is going to break the seed dormancy and it will promote the seed germination process, on the other hand, the ABAs are going to promote the seed dormancy and it will inhibit the process of seed germination. So this is how it works. All right. So those are antagonistic hormones. Next question. The next question says that from his experiment, S.L. Miller produced many amino acids by mixing the following in a closed flask. So this is a kind of an experimental question, which is a kind of a direct question, you can say, that has been put up. And so basically the answer to this question is very, very pretty much clear, that in a closed mixture, in a flask having all the closed mixture of the amino acids, Miller actually produced a combination of some gases, combination of some gases. And out of all those gases, if you will see, the main gases included, and the temperature is actually very important, the gases that were liberated was methane, nitrogen, ammonia, and along with water vapor at 800 degrees Celsius. So this will be the case. All right. Not ammonium. Here also you can see here this is wrong. This is actually methyl that has been given. So again, this is not an option, and this wasn't liberated, not at all liberated. So this was not an option. The left out options are this one and this one, but the temperature variance, if you are going to see to it, then this is 600 and this is 800. So 800 is the correct option. All right. So what is the explanation? So the explanation is that this experiment was actually conducted in the year 1953. So in 1953, Miller created electric discharge. So he actually created a setup. So he created electric discharge in a closed flask containing methane, hydrogen, ammonia, and water vapor at 800 degrees Celsius. So this is again a kind of a direct question. All right. So option D is the correct answer. Clear? Now next question. Next question says that experimental verification of the chromosomal theory of inheritance was done by. Now again, chromosomal theory of inheritance was given by T.H. Morgan, but the entire explanation, if you are going to see, so the experimental verification was actually done by Morgan. All right, Thomas and Morgan only was the person who did it. So this theory was actually, initially it was proposed by Sutton and Bowry. All right, Walter Sutton and Theodore Bowry were the scientists who actually proposed it. But verification was done by T.H. Morgan using the fruit flies. That means Drosophila melanogaster. So we can just write that chromosomal theory of inheritance was proposed by Sutton and Bowry. All right. However, experimental verification was done by Thomas Hunt Morgan, T.H. Morgan. So this is the case. So what will be the correct answer out of all the options? So you can see that Morgan has been given. So option C is the correct answer. Clear? Now, next question is question number 60. Now, question number 60 says that the body of the ovule is fused within the funicle at. So the body of the, if you are going to look into the body of the ovule, so ovule, what is ovule? Ovule, we know that ovule is considered to be as an integumented megasporangium. So ovule is integumented megasporangium until it is not fertilized. All right, so that is the case. So this is also a direct question that is related to the structure of the ovule, if you're going to see. All right, then the part that is going to be fused with the funicle in the ovule, or the body through which the body of the ovule is actually fused in the within the funicle, that is called as hilum. All right, so we can just write that the body of ovule is fused within funicle at hilum. So hilum is basically the point of attachment, you can say. This is the point of attachment where the body of the ovule is going to fuse with the funicle. So that is the case. So if we just draw the figure, have an idea. This is like this. All right. Now, if we just label the different parts, so these are integuments. These are the integuments. Then this part which you can see, this is the structure of an anatropous ovule that I have drawn. This is the nucellus. Clear? Then this point through which the pollen tube will be entering inside the embryo sac. So this is called micropyle. So ovule has basically two ends. All right. So that is called micropyle. Then this part, the opposite part is called chalaja here. Then this part which you can see is called as the hilum part, where the funicle is going to fuse with ovule. And this is the funicle. So this is a kind of a stalk-like structure, you can say, funicle, through which ovule is actually attached to placenta. So this is funicle. And this is the hilum that we have seen. All right, hilum part. So this is the structure for the ovule that we have seen. So the correct answer turns out to be high. Clear? Now, next question. Now the next question says that identify with reference, you have to identify the correct statement with reference to the human digestive system. All right. So if you just look into the figure of the human digestive system, if it is a kind of a picturization that is there in your mind, then you have to identify the correct statement. So correct statements, the first statement says that serosa is the innermost layer of the alimentary canal. All right. So basically serosa is not the innermost layer, but it is the outermost layer, let me tell you. All right. So if you just look into the section of the gut, so serosa because if you look from outer to inner section, then it goes like serosa is the outermost layer, then serosa is followed by mucosa, then serosa is followed by mucosa, then submucosa, and then muscularis. So this is how it goes on. So serosa is actually the outermost layer. Outermost layer of alimentary canal. Layer of alimentary canal. So again, the statement is the wrong statement. Ileum is a highly coiled part. All right. So ileum, so if you people remember, then ileum is a part of your small intestine, and small intestine has been divided into three distinct parts. Small intestine has three parts, you can say. All right. So those three parts are duodenum, jejunum, and ileum. So duodenum, jejunum, It's jejunum and ileum. All right. So ileum is actually the highly coiled part of the small intestine. So yes, this is actually a correct statement. This is actually the highly coiled part, you can say. All right. So this is a correct statement, but let us take a look into the other statements as well. All right. So C statement says that vermiform appendix arises from duodenum. Not at all. Vermiform appendix actually arises from the cecum part of the large intestine. So this is a wrong statement because vermiform appendix, this is a kind of a vestigial organ. So vermiform appendix arises from cecum of large intestine, which is the first part of large intestine. Just like small intestine is divided into three parts, the large intestine also is divided into three parts. That means cecum, colon, and rectum. All right. Then the D option says that ileum opens into small intestine. No. This is also again because ileum is a part of small intestine only. So this will be opening into large intestine, not small intestine. So ileum opens into large intestine. Large intestine. So the correct option turns out to be option number 2 over here. Clear? That is the case. Let's move on to next question, which is question number 62. Now, dissolution of the synaptonymal complex occurs during. See, synaptonymal complex is basically a complex which is formed during the process of synapsis that occurs in zygote. All right. So formation of synaptonymal complex, you can say, is in zygote. Formation of synaptonymal complex, which is called bivalent or tetrad synaptonymal complex, that takes place in zygote. And this is via synapses only. Huh? This is via synapsis. This occurs in zygote. But the dissolution of the synaptonymal complex, that is going to occur in the fourth phase or fourth stage of the prophase 1, and that is going to be the diplotin stage. That means this one. All right. So again, this is not an option. Leptotin is the very first important phase where the chromosome condensation takes place. So you can say that this is the first option. Then diplotin is the option where the dissolution of synaptonymal complex occurs. So this is characterized by dissolution of synaptonymal complex. And all these, we know that all these are the phases of prophase 1 of meiosis 1. Of clear? Now, the C option will be leptotin. So leptotin is a phase where chromosome condensation occurs. This is also called as bouquet stage. So chromosome condensation occurs. All right. And the D stage is the pecitine. Pecitine is actually characterized by crossing over stage. Pecitine is a crossing over stage. So the correct answer is diplotin. All right. So this is the case. Next question. Question number 63, which states that Floridian starch has structure that is similar to. All right. Now, Floridian starch, let me tell you that Floridian starch is one of the kind of a reserve food material of red algae. All right. So, Floridian starch, this is actually a modified form of starch, you can say, or a complex form of starch. So, is a reserve food material in red algae. And this Floridian starch is similar to the structure of amylopectin and glycogen here. All right, because mannitol is different. This is mannitol and laminarin. Mannitol and elgin, this is found in case of your brown algae. This is the reserve food material in case of brown algae, you can say. Clear? Then laminarin is also found in case of brown algae. Starch and cellulose is being found in case of green algae. All right. So that's why it has a similar structure which is equivalent to amylopectin and glycogen. So its structure is similar to amylopectin and glycogen. Clear? This is the case. Now, next question is question number 64. So in the question number 64, it says that the specific palindromic sequence, which is recognized by ECO-RI. So palindromic sequence, this we have already discussed it before also. So palindromic sequence means the sequences. Palindromic sequences means the sequences which are, you know, yeah. So palindromic sequences are the sequences which you read from, you know, whether from the your forward direction or whether from your backward direction, all seems to be similar. All right, like Malayalam, if I say. So ECOR1 ECOR1 is basically a kind of a restriction enzyme that recognizes a specific palindromic sequence, if I tell you. All right, so for that part, if you are going to see, then the correct option out of all the options, this is very specific because for each bacteria and for each enzyme specifically, then, you know, for not for bacteria, but for the enzyme that has been isolated from the particular bacteria, which is E. coli, that restriction enzyme is very specific for cutting or cleaving the, you know, bases. All right. So here the correct sequence will be, if you are going to recognize, that means that is being recognized by ECOR1. So this is G double A T T C. All right. 3 prime. And that means option number D I'm talking about. And here will be 3, then C, T, T, A, G. All right. So this is the case. So this is the specificity where they target, and where they target, they are going to target over here. So the cleavage site is over here, which you can see. This is the, you can say, cleavage site. Cleavage site or site of restriction. Restriction. This here also the restriction site will be here. which is going to be recognized by the EcoR1, recognized by like this. Clear? So this is the case. So out of all the options, this is very specific. This has to be learned because for every restriction enzyme, the sequences are very specific where they need to be cut. So that's why the option D is the correct answer. Clear? Now, next is the question number 65. Now, in case of the question number 65, it says that you have to select the correct match with the kind of the trait that is being possessed. So here are the diseases which are being disorders actually which are being given over here. All right. So if you will see, then the first option is the phenylketonuria. Phenylketonuria is actually an example of autosomal recessive trait, but not the dominant trait. So phenylketonuria. So phenylketonuria, this is an autosomal recessive trait. Clear? So this is not the correct option. Then there is sickle cell anemia, which is autosomal recessive trait, and and this is actually present on the chromosome number 11. So again, this is also a correct answer. This is the correct answer. It is actually an autosomal recessive trait with that, that is relatable to chromosome number 11. So sickle cell anemia. This is autosomal, where the RBCs, they changes into sickle shape. From the normal biconcave shape, they changes into sickle shape. Autosomal recessive trait. So this is actually the correct statement. Then there is thalassemia. So thalassemia again is a kind of a blood disorder only, and thalassemia. And thalassemia is a category of, this is an autosomal recessive trait, but not a Y-linked trait actually. This is also an autosomal recessive trait. So again, this is not the correct option. And the last turns out to be hemophilia. Now hemophilia is X-linked recessive trait. Hemophilia. Where this is one such disease where there is a disorder in the blood clotting process. This is a blood clotting disorder, you can say. So hemophilia is a X-linked recessive trait. That means this is a category of sex-linked recessive trait, which is passed on through sex chromosomes, but not from the autosomes. Clear? This is the case. So out of all the options that we have seen, so the option B, that means sickle cell anemia, is the correct match. All right. Okay, so let's talk about the next question, which is question number 66. So you have to identify the substances that have the presence of glycosidic bond and the peptide bond. All right, that means two of the bonds are being given and you have to identify it in the respective structure. All right, so if you just look into the option, the first option says, is it glycerol and trypsin? The second option says to be the cellulose and lecithin. Then it is inulin and insulin, and that is chitin and cholesterol. All right. Now let me tell you that let me just give you a brief idea about the glycosidic bond. So glycosidic bond is basically found in the category of this C, which is the correct answer is going to be the option C, which is inulin and insulin. And the reason for this is because inulin is actually it's a polysaccharide of fructose. All right. So inulin, we can say, is a polysaccharide of fructose. It's a polysaccharide of fructose. All right. Which is commonly also known as fructan, which is also considered to be as fructan. Now these fructose units, all right, so we know that fructose is a monosaccharide. So these fructose units, since this is a polysaccharide of fructose, so all the fructose units, they are linked to one another through the process of glycosidic bond. So you can say the fructose units are linked via glycosidic bond. Glycosidic bond. So this will be the reason. All right. Now regarding the insulin, if you are going to see, so insulin basically is a kind of a proteinaceous hormone. All right. So this is a category of proteinaceous hormone, and we know that in case proteins are what? Proteins are actually the polymers of amino acids. All right. And those amino acids in turn, they are linked to one another through peptide bond. All right. So we can say that insulin is a proteinaceous compound. So insulin is a proteinaceous compound, and since this is a proteinaceous compound, so this is again also a kind of a complex polymer, because it consists of around 51 amino acids. So this usually consists of 51 amino acids, and all these amino acids, they are linked to one another through peptide bond only. All right. And these amino acids are linked together through peptide bond. All right, so this will be the reason. Clear? So the correct answer is going to be the inulin and insulin. All right. However, other molecules, they are having different kind of bonds that are found in them. All right. So let's move on to the next question, which is question number 61. Now the process of growth is maximum during. So there are certain phases that has been given over here, and it is talking about the, this is actually a question related to the growth phase, process of growth and development. So if you just look into the graph, if you just, so the graph generally turns out to be, this is a graph which is plotted against, growth, against time. So basically, if you just look into the graph of growth, so this goes like this. So this basically gives out a figure of sigmoidal growth, which we observe it. And there are various phases. If you remember, this is the first phase, then this is the second phase, then this is the third phase, and this is the fourth phase of the graph. All right. So here is basically growth, and this is in relation to time. This. All right. So this is the case, and this is usually observed in case of plants, if I'm talking about. So if you look into the various phases, so this phase basically represents, that means the first phase represents the lag phase, which is usually a phase of slow growth. This is a phase of slow growth, because, or you can say the growth is being just started or initiated, you can say. Then this phase is called as the second important phase, which is called as the log phase, which is also considered to be as exponential phase. So this is also known as log phase or exponential phase. And this is one such phase which shows the maximum rapid growth, you can say. Rapid or maximum growth, you can say. That is observed typically. And then this phase is this third phase basically represents this one. This is, or we can just label it like this. So this phase basically represents the, we can, yeah. So this phase basically represents the deceleration phase. Deceleration phase is basically the declining phase, you can say. So that's why this is called deceleration phase, or the phase of, or declining phase. And this last phase is considered to be as the stationary phase, or the phase where there will be further no growth. So this is stationary phase. All right. So this is the graph that is usually being plotted. And this is actually S-shaped growth curve that you are seeing it. Just a second. This is S-shaped growth curve. So out of all the phases that we have seen, log phase, which is also considered to be as exponential phase, that shows the maximum growth here. That means option D is going to be the correct answer that you can see. All right. However, this is a slow phase. Again, this is not an option. Senescence and dormancy, these are completely two different things. So these are actually a process of development, but not a part of the growth. So definitely this won't be, these two also won't be an option. So option D is the correct answer. All right. Now moving on to next question. Now question number 68 is the match the column kind of a question that has been asked. And you have to match the column. There are certain structures of year that has been given over here, and in the column 2 with their respective functions or localizations that has been given. All right. Now, so if you just look into the question, or the categorization, if you're going to see, then the first basically relates to the organ of Corti. All right. Now, organ of Corti, if you're going to see, so organ of Corti is correlatable to the second option. Out of all the options that you can see, it connects middle ear and pharynx, then coil part of labyrinth, then this is attached to the oval window, then that is located to the basilar membrane. All right. Now, so organ of Corti is one such organ, if you people remember, then this organ is actually located on the basilar membrane. All right. So this is a structure which which is actually located on the basilar membrane. So we can say that organ of Corti is relatable to located on basilar membrane. So this is the localization. Now the second option says cochlea. So cochlea, if you people remember the structure of cochlea, this is actually 2.5 turns of cochlea, that's why considered to be as the coiled part of labyrinth. So option B is going to match up with this one. So this is going to be with A and this is with B. Clear? So cochlea is the coiled part of labyrinth. Clear? Now, the option C says eustachian tubes. Eustachian tubes is actually going to connect the middle ear cavity with that of the pharynx. So if you just look into the two options which are left, so this is the C option which is going to be matched up over here. So eustachian tube, now this eustachian tube is going to match up with, or this is going to connect middle ear with pharynx. And the last option is the stapes. So stapes, if you people remember, these are the three muscles that are found. So malleus, incus and stapes. So stapes is the innermost part that attaches to the oval window. So this is basically the ear ossicles, as you can say. So this is attached to oval window of cochlea. Clear? So this is going to match up with the option. So now let us take a look into the different options that has been given. So out of all the options that you can see, A is going to match up with 4. So A is going to match up with 4. B is going to match up with 2. Then C is going to match up with 1. D is going to match up with 3. All right. So with this, we come to know that option B is the correct answer. All right. So this is the case. Now let's move on to the next question, which is question number 69. So question number 69 says, the process which is responsible for facilitating the loss of water, and that too it is in liquid form. Remember, from the tip of the grass blades at night and in the early morning. So that is because of, that is called root pressure or imbibition or plasmolysis or transpiration. All right. Now, out of all the option, if you just remove out the options which doesn't matches to this point, so imbibition and plasmolysis won't be an option, because imbibition is basically the absorption. It is the process of absorption of the water through the seed coat. All right, or via seed coat you can say. And plasmolysis is an entirely different thing, in which there is a loss of water from the cell, because of which the cell is going to shrink. All right, so only the two options left is root pressure and transpiration. Now, if you just look into the question, so the question is asking that it is in liquid form. Liquid form basically means this is in form of water. And in case of transpiration, if you are going to see, there is loss of water in form of water vapor, but not in the form of liquid. In form of water vapor from aerial parts of plants. All right. So this is the definition. So this is actually in the form of water vapor, but here it is asking about in the water. So the correct answer is going to be the root pressure over here. All right. Because root pressure is a kind of a positive hydrostatic pressure that develops in the tracheary elements of the xylem, and because of that reason only, because of that root pressure only, water comes out in the form of liquid from the tip of the grass blades. So we can say that root pressure is a positive hydrostatic pressure that develops in the tracheary elements of xylem, leading to water loss in liquid form from tip of grass blades. All right, so this will be the reason. All right, now let's move on to the next question, which is question number 70. Now question number 70 says that you have to identify the wrong statement with respect to immunity. Wrong statement, that means the statement which is incorrect, we have to identify it. So the first statement says that when ready-made antibodies, they are directly given, it is called as passive immunity. All right. So we have seen that immunity is of two types. It can be either active or it can be either passive. So definitely passive immunity is such immunity, that is given in the form of pre-antibodies, you can say. All right. So that is the case. So this is a correct, basically statement. This is the correct statement. This is not the wrong statement. So this is actually the correct statement. Clear? Now, active immunity, the second option says that active immunity is quick and gives full response. All right. So is it that so? I mean, like is active immunity quick and gives full response? So let us take a look in the other options as well, and then we'll decide it. So the next question statement says that fetus receives some antibodies from mother is an example of passive immunity. Again, this is also a correct statement, because if you people remember, then fetus is going to receive, just a second. Fetus basically receives the preformed antibodies from the mother, and that is through milk feeding process. And that antibody that has been released from the mother, that is called as IgA antibody. So this is an example of passive immunity. So this is a correct option exactly. All right. And the next is that when exposed to antigen, either living or dead, antibodies are produced in the host bodies, and this is called active immunity. All right. So again, this is also a correct statement, because yes, correct statement, this is one such method. That means active immunity is one such process when a body gets exposed to any kind of antigen or antigen antibodies antigen, then in response to those antigens, the antibodies are actually produced in the host bodies itself, you can say. So this is again a correct statement. All right. Now this statement, which says option B is only left, which says that active immunity is quick and full and gives full response. Now this is the wrong statement. Why this is a wrong statement? Because active immunity is one such process which is a slow process. Since it is being activated from inside the body, from itself, so that's why it takes a little time to get activated, and it takes time to give its full effective response. in comparison to the passive immunity, where the antibodies which are directly given to the body, that gives a quicker response. So that's why we can say that active immunity is slow and takes time. So this is slow and takes time to give its full effective response in comparison to passive immunity So in comparison to passive immunity, where preformed antibodies are administered. All right. So that's why the wrong statement is going to be this one. And the correct statement will be option number B over here. This one, with regard to the wrong statement that we are talking about. All right. So option B is going to be the correct option with regard to wrong statement. Question number 71. Now, in case of question number 71, it says that in water hyacinth and water lily, the pollination takes place through. All right. So the options are water currents only, wind and water, insects and water, and insects or wind. Now, let me tell you that though this water hyacinth and water lily, this is actually an exceptional category of aquatic plant. Water hyacinth and water lily. These are actually an exceptional category where though being, these two, though being aquatic plants, these two are such category of aquatic plants in which the process of pollination takes place either through insects or through wind, but not by water. All right. So pollination is done by insects or wind. And why is it so that why the pollination is being done by insects or wind? Because these flowers, the flowers of these plants, all right, they emerges above the level of water. This is because, or this is due to emergence of flowers of these plants above water. And that's why this gives an opportunity to the insects also and to the wind also to carry out the process of pollination. All right. So the correct answer out of all the options is option number D, which is insects or wind. All right. Now, next question is question number 72. Question number 72 says that which is the important site of formation of glycoproteins and glycolipids in the eukaryotic cells. All right. Now, this is again, we can say this is a kind of a direct question because it is directly asking about the, function. This is actually the functional aspect of the cell organelle, and given below are the listed cell organelles, as you can see. So the site of formation of glycoproteins and glycolipids is basically the Golgi bodies, because if you people remember, then Golgi bodies are actually responsible for glycosylation of proteins and lipids. So that's why. So the correct answer will be that Golgi bodies, because we know that polysomes are actually the you can say the combination of ribosomes or collection of ribosomes. Polysomes means that when several or multiple ribosomes, they are wounded to one another, so that is called polysome. This won't be an answer. Endoplasmic reticulum, we know that it is related to your it is either rough or it is either smooth. So this won't again be an option, because rough is involved in case of protein synthesis, while smooth endoplasmic reticulum is involved in lipid synthesis and detoxification of cells. And again, peroxisome is related to beta oxidation of fatty acids. So this won't be an option. All right. So that's why Golgi bodies are the site of formation of glycoproteins and glycolipids. So they're the site of formation of glycoproteins and glycolipids due to glycosylation being performed. In them. So option B will be the correct answer. Clear? Now, next is question number 73. So question number 73 says that which of the following regions of the globe, entire globe it is talking about, exhibits the highest species diversity? So the highest species diversity it is talking about. So the options given are Madagascar, Himalayas, Amazon forest, and the Western Ghats of India. All right. So if you just consider the different options, then out of all the options, 70%, almost 70% of our global species diversity is being contributed by Amazon forest. All right, which is localized in the South American zone of the globe. And it has the greatest biodiversity on earth. So this is also a kind of a direct question, we can say. In India, if we consider, then it is, since here it is talking about the entire globe, so this we have to keep this in mind, because in India there are Himalayas, then Western Ghats also, which are considered to be as the hotspots of India. But if we just consider the hotspot of the entire globe, then Amazon is being there, Madagascar is being there. But highest species diversity, if you just look into this parameter, then it is none other than the Amazonian rainforest, you can say. So the answer will be the Amazon forest. Amazon forests in South America. Has the highest species diversity. On earth. And not only in terms of animals or plants, but also in terms of all the insects, all those categories of invertebrates, vertebrates and everything. So that's why they are considered to be as the most diversified zone that is found in the globe. Clear? So option C is the correct answer. Now moving on to next question, which is question number 74. Now question number 74 says that the goblet cells of the alimentary canal, they are modified from, all right. So goblet cells, basically goblet cells are, these are the cells that are found in the alimentary canal, and they are actually the modifications of these cells are primarily, the functions of these cells is primarily to release or to secrete mucus, all right. And this function, in order to release mucus or, secrete mucus in the alimentary canal, they are actually a modification of columnar epithelial cell. So there won't be an option of chondrocytes or compound epithelium or squamous epithelium. All right, so this is again a kind of a direct question, you can say, that has been asked. So we can say that goblet cells of alimentary canal are modified from columnar epithelial cells. And why they are modified? So they are modified to secrete mucus, which is of lubricating nature. All right, so the option A is the correct answer. Now, next question is question number 75. Now question number 75 says that which of the following is correct about viroids? All right, so there are certain statements that has been given regarding the viroids. So viroids is basically a kind of, you can say, virus particle, but that is not having any kind of protein coat. See, the difference between virus and viroids is that viruses, they are the particles that have the presence of proteinaceous spot around themselves, if you look into the structure. But these are actually, their RNA are actually free from any kind of a protein coat, and they have the smallest, their RNA is also small, and that's why it is called as naked RNA. So the first option, if you look into the first option, so that probably looks to be the correct answer, because they have free RNA. Free RNA basically means naked RNA, you can say. So free RNA means naked RNA. That means without any kind of protein coat. That is without capsid, you can say. So this is the correct statement that, that seems to be. But let us look into the other options as well. They have DNA with protein coat. First of all, they do not have DNA as the genetic material. So this option and this option won't be an option. And they have RNA with protein coat. This is a correct statement for virus, not but for viroids. All right, so that's why the correct statement turns out to be the option number A. All right. So they actually cause various kinds of diseases, and one of the most prominent diseases that has been discovered is by Theo Diner, and in 1971, and they usually cause, the first disease to be discovered is potato spindle tuber disease. So they have, the discoveries are still going on, the research is still going on regarding them. Now, the next question is that secondary metabolites such as nicotine, strychnine, and caffeine, they are produced by plants for their, all right. Now, all these secondary metabolites, which are nicotine, strychnine, and caffeine. Now, let me tell you, this is a question that has been asked as a kind of a question from the, in case of your population interaction. And secondary metabolite, as already I told you and discussed with you, that secondary metabolites are those metabolites that are not released through their entire life cycle, but at only specific stages of their life cycle when it is required only. That's why. All right. So if you look into these, options, strychnine and then caffeine and then nicotine, so are they responsible for their growth response? No, because as I told you, if they will be responsible for growth response, so they should be produced, indefinitely, but they are not. Effect on reproduction? No. They have no relation with that of the reproduction, and nutritive value? No. They have no relation with the nutritive value. All right. So the only option left is the defensive action. And these metabolites, they are actually produced by plants as a kind of a defensive mode or as a kind of a defense. Why defense? So that they can be protected from any kind of grazing by the grazers and the browsers. So you can say that these chemicals means all these above mentioned chemicals, you can say, are released by plants as defenses against grazers or browsers. So they are actually being released by plants as defenses against grazers and browsers. Because once the grazers and browsers, they try to eat those plants, these chemicals are introduced into their body and they are going to harm their internal structures, or they can also kill them as well if the intake is very high. All right. So that's why the correct answer is option number B. All right. Now let's move on to the next question, which is question number 77. Now here again, the match the column kind of a question has been given. And in the column 1, you can see the different, categories of the different options that has been given, and column 2 has another respective options. So option number A says placenta. All right. So if you just look into placenta, so we know that placenta is actually the structural and functional unit between a mother and a fetus. All right. However, placenta is also considered to be as a temporary endocrine gland. So this is also known as temporary endocrine gland. And if it is a temporary endocrine gland, so this during the process of pregnancy is going to produce a very important hormone, and that hormone is called as HCG, which is human chorionic gonadotropin hormone. So A option is going to match up with B, this one. Sorry, this is going to be A over here, A. Clear? This is the case. Now what about B? Now the B option says it to be the zona pellucida layer. Now zona pellucida is one of the important layers of the ova. This one. Zona pellucida is considered to be as one of the important layer of the ovum because this is a primary egg membrane, you can say. So zona pellucida is a primary egg membrane secreted by secondary oocyte. This is basically a layer of ovum, you can say. Secondary oocyte. Clear? So this turns out to be for option B. Now the third option says bulbourethral glands. Bulbourethral glands are one of the accessory glands that is found in case of your male reproductive system, and that helps in the lubrication of penis. So this will be for C. All right. So bulbourethral glands. Urethral glands are the male accessory glands. Accessory glands that helps in lubrication of penis. Lubrication of penis. Yeah, this is this. And the last option, which is option number D, is left with Leydig cells. So Leydig cells, which are also known as interstitial cells, so they are responsible for secreting. They are one of the important cells which are found inside the section of the seminiferous tubules that you can see. This is basically related. This Leydig cells, they are basically related with the secretion of androgens. That means your testosterone hormones in case of males. All right. So Leydig cells synthesizes and secretes also. Testicular hormones or androgens. Clear? So this. Now let us look into the different options that has been given. So A is going to match up with option number 2. A is going to match up with option number 2 over here. Then B is going to match up with 3. Then C with 4 and D with 1. So out of all the options, C is going to be the correct option. Clear? Now, next question. The next question says that ray florets have. Now again, this is a kind of a direct question that has been given. So there are two kinds of florets. So ray floret and disc florets. All right. So ray floret basically have, and this is found in case of sunflower. Sunflower is one of the examples of ray floret and disc floret basically. So ray florets usually have inferior ovary. That means they are epigynous flowers, which are, in which they are found, and generally in the members of Asteraceae, and one of the best example is sunflower. So it will be the inferior ovary that is going to be the answer. That means option number D. So you can write the reason that ray florets have inferior ovary. So ray florets have inferior ovary, and if the ovary is inferior, then definitely this indicates what? Epigynous flowers. So epigynous flowers are formed in family Asteraceae. Which is the largest family, which is considered to be as the largest family of the angiospermic plants. And one of the best example of ray floret and disc floret includes sunflower. All right, so this is the case. Now, next question is question number 79. Now, in case of the question number 79, they say that which of the following is put into anaerobic sludge digester for further sewage treatment. All right. So for further sewage treatment, which of the following category is put into the anaerobic sludge digester? Now, the options are floating debris. So if you people remember that during the process of sewage treatment, there are three stages: primary, secondary, and tertiary. And this removal of this anaerobic sludge digesters, they are used in the secondary treatment process, but not in the primary treatment. So floating debris won't be an option, because this floating debris is actually removed in case of your primary treatment process. All right. So that is the case. And the second option says to be the effluents of primary treatment. So effluents of primary treatment won't be an option, because these anaerobic sludge digester is a part of secondary treatment. Remember, this is a part of secondary treatment. And if there is a part of secondary treatment, then this won't be an option. Clear? Now the two options which are left is the activated sludge, and along with that there is primary sludge. Now let me tell you about activated sludge. So activated sludge is basically the sediment that is going to settle down in the settling tank, you can say. So that is called activated sludge. So out of that activated sludge, a small part of the activated sludge is pumped back into the aeration tank, while most of it is digested. Most of the part of the activated sludge is being transferred into another tank, and that tank is called anaerobic sludge digester, where they will be undergoing the process of degradation or digestion, you can say. So out of all these two options that is left, this will be the correct option. So the statement will be that the sediment, let me tell you about first the definition. So the sediment in the settling tank is called activated sludge. Now this activated sludge has two portions. Sludge, clear? Now it is made into two portions actually. So made into two portions. One is a smaller portion and the other is a bigger portion. One smaller and other major portion or bigger portion. Now this smaller portion is considered to be as the activated sludge only. This is passed back to aeration tank, where it will continue the process of sewage treatment for the cycling. It's passed back to aeration tank. All right. While the bigger portion, this remaining part of the activated sludge is pumped back to large tank. So this is passed to large tanks for anaerobic sludge digesters. Anaerobic sludge digesters, where they are digested by the methanogenic bacteria. All right, so this will be the case. So that's why the correct option will be option number C. Clear? Next question. Next question is question number 80, and this says that the enzyme enterokinase helps in the conversion of, all right. So this enzyme enterokinase is, let me tell you that this enzyme is actually responsible for, this is actually being released or secreted by the intestinal mucosa. All right. And once these are released, these are being released by the intestine, this enterokinase, it is actually going to convert the inactive trypsinogen into trypsin. All right. And once this, and this trypsinogen is actually a zymosin that has been released from the pancreas. So enterokinase actually helps in converting the inactive trypsinogen into the trypsin part. So you can say that trypsinogen, which is an inactive enzyme, this will be converted into trypsin, which is an active enzyme, with the help of an intermediate enzyme, which is enterokinase. Now, this enterokinase, as already stated, that this enterokinase is secreted by intestinal mucosa and converts it into active trypsin. And this is being released from pancreas. So this is a zymosin from pancreas. All right. So the correct answer is going to be trypsinogen into trypsin. That means option number A. All right. Now, the next question is the question number 81. Now this question number 81 says that, again, this is a match the column option that has been given, which is again very direct, that can be done. So this is relatable to your animal kingdom that has been asked. All right. So here, if you're going to see, then it is talking about the column 1. So column 1, the first option is says gregarious polyphagous pest. Now the gregarious polyphagous pest is called as locusta, which is a member of, insecta phylum. All right. So option number A, that means locusta. This is a gregarious pest. A gregarious pest. So this means this will be correlatable to A option. Clear? Now, adult with radial symmetry and larva with bilateral symmetry, if you're going to see. So undoubtedly, this is going to be, this is a feature of echinodermata, and this is going to be correlatable to your, in case of echinoderms only, the adults are having the radial symmetry and the larva is having the bilateral symmetry. So that's why this is relatable to Asterias, which is this one, option number 1. All right. So here will be option B. Yeah. So Asterias, which is an echinoderm. Sorry, it's echinoderm. Echinoderm. So its adult has radial symmetry, while larva has bilateral symmetry. Clear? Now, option number C says book lungs. Now, book lungs is one of the important features of scorpion. Again, This is a example of phylum Insecta. So C over will be here. So scorpions respires through booklets. Sorry, it's respires through booklets. And then option D, bioluminescence property. Bioluminescence is basically one of the property of the living organisms to emit light on their own. That is called bioluminescence. And this is the characteristic feature of ctenophora members. And the example is ctenoplana. All right. So this is the D option that has been given. So bioluminescence is well marked in ctenophores. So this is actually well marked in ctenophores. All right. So this will be the case. So let us take a look into the different options. A is going to match up with option 4 here. Then B is going to match up with 1, C with 2, and D is going to match up with 3. So the correct option is option number A. All right. Now, next question is question number 82. Now, question number 82 says that the presence of which of the following conditions in urine are indicative of diabetes mellitus. All right. So diabetes mellitus, basically, this is again a kind of a direct question that has been asked. So there are ketone bodies. If the ketone bodies are found in case of the, in case of urine, then this is an indicative factor that, there is presence of, or there is an indication of diabetes mellitus. So ketone bodies also, which is called as ketonuria, and if there will be the presence of glucose also, which is called as glycosuria, then both the conditions are responsible. So out of all the options that you can see, uremia and renal calculi won't be an option. This is hyperglycemia and renal calculi. Again, this won't be an option. Uremia and ketonuria won't be an option. Yeah. So our answer will be option number D over here. Clear? So you can say that presence of ketone bodies. Now, so if there is presence of ketone bodies in urine, that is called ketonuria. And glucose in urine, which is called glycosuria, is indicative of diabetes mellitus. Diabetes mellitus. Yeah, this will be the case. Now, so option B is the correct answer. So let's move on to next question, which is question number 83. Now, question number 83 says that, again, this is a match the column question. Column 1 has been given along with column 2. So column 1 says floating ribs have been found. All right. So floating ribs are basically, you can say floating ribs are basically called as to be as the, this is, you can say, floating ribs are those ribs which actually doesn't connects to the sternum part. That means they do not connect, they are not connected ventrally, you can say. So out of all the ribs, in the humans that is being found, so floating ribs are actually the 11th and the 12th pair of ribs that actually doesn't connect ventrally to the sternum. And that's why they are dorsally connected, but they are not connected ventrally. So that's why they are called as floating rib, because they are floated from one side. They keep floating from one side. So that's why option A, which is floating ribs. So floating ribs so ribs which are 11th and 12th pair are not connected ventrally. So they are not connected ventrally by sternum. And for that is the reason that is called as floating. So this will be with A option. Then the next option is acromyon. So acromyon is basically, you can say acromyon is a kind of is a flat expanded process of the the spine of the scapula. All right. So that is considered to be as the, or in simple language we can say that acromyon is actually the lateral end of the clavicle, which which articulates with the acromyon process. So that's why it is going to go with option 3 over here, which has been out of all the options that is given. So this will be with B option. So you can say that acromyon is the lateral end of clavicle. End of clavicle, which articulates with acromyon process. Yeah, this will be the case. So this is a flat expanded process of spine of scapula. Now the third is scapula. So scapula is located, if you just look into the options, so scapula is once a structure which is located in the 2nd and the 7th ribs. All right, so this is again a kind of a flat triangular bone, you can say. So scapula is a flat triangular bone in the dorsal part of the thorax. Between 2nd and 7th rib. So that's why option A. Sorry, this is with option number 1 actually. Here will be the correct answer. This is with C. And the last option is glenoid cavity, which is relatable to the head of the humerus. So glenoid cavity is again it's a cavity of scapula, which articulates with the head of the humerus in order to form the shoulder joint. Right. So option number D says that glenoid cavity. So glenoid cavity of scapula, you can say, articulates with head of humerus to form shoulder joint. Clear? So that now let us take a look into the options. So A is relatable to fourth, B is relatable to third, then C to 1 and D to 2. So the correct option will be option number C over here. All right. Now, let's move on to next question. Now, this question says that how many true breeding pea plant varieties did Mendel select as pairs? All right, which were similar except in one character with contrasting traits. All right. So basically, if this is a kind of a direct question, again, you can say, because this is a question that is relatable to your plant genetics part. And if you just remember, so Mendel actually selected, 7 different pairs of contrasting traits. So select as pairs it is talking about, and how many, varieties it is talking about. So since Mendel chose pea plant for breeding, all right, and in these pea plant, he basically chose or selected, you can say, 7 different pairs of contrasting traits. Different pairs of contrasting traits. So if it is 7 different pairs of contrasting traits, then making it a count of 14 in total, you can say. Making it a count of 14 in total. So that's why the correct option will be option number B, which were true breeding pea plant varieties. All right. Now, next question is question number 85. Now, the question number 85 says that if head of the cockroach is removed, then it may live for a few days because. All right. So this is a question that has been asked from your animal organization, and that too from the animal type cockroach. So now if head of the cockroach is removed, it may live for a few days because the option A says that the cockroach does not have nervous system. Then the second says that the head holds a small proportion of a nervous system while the rest is situated along the ventral part of its body. Then the head hole. Now see, the first option, let me tell you, let us just move on with the options first. So the cockroach does not have a nervous system. This is actually a wrong statement, because cockroaches do have a nervous system. All right, because they have the presence of a though their ventral system, nervous system is not very much developed, but yes, they do have a nervous system. So this is actually a wrong statement. Now the second option we have already read out. Then just let us take a look into the third option, which is the head holds 1/3 part of the nervous system while the rest is situated along the dorsal part of the body. Now let me tell you that the nervous system of cockroach is always ventrally localized. Nervous system of cockroaches ventrally localized. So here it is talking about the dorsal part of the body. So no, this won't be an option. Now the supraesophageal ganglia of the cockroach are situated in the ventral part of the abdomen, if you're going to see. So again, this won't be an option. All right. Now the correct option will be this one, because since the head holds a very small proportion of the nervous system, while the rest, that means major part, rest means the major part of the nervous system is actually situated along the ventral part of the body. And for that reason, if the head of the cockroach is removed also, then it may live, or it might live for few days. All right. So option B is going to be the correct answer. This is also wrong. All right. Now, next question. Now the next question says that which of the following refer to the correct examples of organisms which have evolved due to changes in environment brought about by anthropogenic action. Remember, anthropogenic action means what? Anthropogenic action means the action which is done by humans, or in involvement with man, you can say. So anthropogenic action it is talking about, which are actions by humans involving humans. All right. Now you have to look into the different examples that has been brought due to the changes in the environment. Now Darwin finches of the Galapagos Island. Now Darwin finches of Galapagos Island, this is an example of natural selection, not an example of anthropogenic. All right. So this won't be an option that can be included actually, because this option is an example of, so this is an example of natural selection and adaptive radiation. Natural selection and adaptive radiation. So definitely this won't be an option. So then it's option B, which is herbicide resistant weeds. This can be an option because herbicide, actions, giving of herbicides, or, spraying herbicides in the weeds, that is going to resist the weedy plant, in their spread and reproduction. So this is actually an example of, you can say, man-made created action. So that's why this will be an example. Then there is drug resistant eukaryotes. Again, this is also a man-made kind of, or, it is an action that has been brought about in the changes in the environment because of the introduction of drugs, making the eukaryotes resistant to it. All right, so drug resistant eukaryotes. And then man created breeds of domesticated animals like dogs. Again, this is also a kind of a man-made created kind of a breeding, you can say, which is anthropogenic breeding, you can say. So which is being done by man. So this also will be involving the anthropogenic action. So with all the options that we have seen, so B, C and D are the correct options. So if you can see over here, then it comes out to be here. Option number B, which is B, C and D. All right. Now next question. Next question is question number 87, which says that you have to identify the basic amino acids from the following. All right. So out of all the amino acids, you have to categorize the basic amino acids. So now let me tell you, so the first option won't be an option because glutamic acid, as the word is suggesting, this is not base, but this is an acid. All right. So this is an acidic amino acid. So glutamic acid is an acidic amino acid. Clear? Then option number B says lysine. Now, before moving on to lysine, let us take a look into the other options as well. Then there is valine and then there is tyrosine. Now tyrosine and valine. Valine is basically a neutral amino acid. This is neither acidic nor basic. So this is actually option number C that has been given. So this is a neutral amino acid. And regarding tyrosine, if we are going to see, then tyrosine is basically an aromatic amino acid. So this is there are two categories of amino acids which are aromatic. So tyrosine, tryptophan and phenylalanine, basically. 3. So tyrosine. So tyrosine is an aromatic amino acid. All right. So the last option which is left is going to be the option number B, which is lysine. All right. Because lysine is a basic amino acid. So lysine is a basic amino acid. So the correct option is option number B over here. All right. Now, next is question number 88. Now, question number 88 says that in the process of gel electrophoresis, the separated DNA fragments can be visualized with the help of. Now, this is a very direct question that has been asked from your biotech chapter. And there has a statement that has been given in your NCRT also, that in the process, gel electrophoresis is basically a process in which the DNA fragments are actually separated from one another using the electrical field methods, all right? And those DNA fragments, they are actually present in the gel-like structure called as agarose gel. And then they are given electric current, and with the help of electric current and depending upon the kind of molecular weight, they get separated. So the smaller the fragment, the farther it moves in the gel. All right, so that is the principle. So basically, once the DNA fragments, they are separated, so their visualization is done under a technique or using, by staining it through a gel or an agent which is called as ethidium bromide. This one, ethidium bromide is which is also called as ETBR. Ethidium bromide is also called ETBR, and that too, once after the staining is done, they can be visualized only and only under UV radiations. All right, so two options are given, either in ethidium bromide in infrared radiation. No, this won't be an option. And these two again won't be an option. So it can only be visualized under UV radiation, and once the visualization is done, so it usually appears in the form of orange bands. All right, so the answer will be ethidium bromide. So ethidium bromide in UV radiation gives an appearance of orange colored separated DNA bands. Just a second. Oh, sorry, just a moment. I'll just write it again. So basically, you can say DNA fragments can be visualized as orange bands. Orange bands. Once it is stained with ethidium bromide, which is ETBr, and under UV radiations. All right, so this will be the case. So this won't be an option, and this is going to be the option A is going to be the correct option. Clear? Now, the next question is question number 89, which says that the sequence that controls the copy number of the linked DNA in the vector is termed as. All right. So that particular sequence, which is going to control the copy number, that means its number of formation of the recombinant DNA that is linked to the DNA in the vector, that is called as. So this is basically the origin of replication site, which is also known as ORI site. All the options that you can see, so there are palindromic sequence, this won't be an option because palindromic sequence is an entirely different thing, which is used for restriction site. So this is basically used for recognizing the restriction site. So restriction site, it is used basically in the recognition of restriction site. And then there is recognition site. Again, this won't be an option, which is this one only. And then there are selectable markers. So selectable markers are used for removing out any kind of the sequences in the DNA from the DNA which are not useful. So those can be used as selectable markers. So this again won't be an option. So you can say that origin of replication which is also called as ORI site, controls the copy number of linked DNA in vector. Why? Because this is actually responsible for initiating. This one is actually responsible for initiating the replication process. So ORI initiates replication process. So this actually initiates replication process. So that is the reason. Clear? So this is the case. Now let's move on to the last question, which is question number 90. Now question number 90 says that the roots that originates from the base of the stem, they are called as. Now this is one of the categories of root that we have already studied. So roots that actually originates from the base of the stem, that are considered to be as the fibrous root. All right, fibrous root, because prop roots are basically the supporting roots which are found in case of banyan. Primary roots are those roots which are found in case of dicot plants, all right, dicots of the angiosperms, and fibrous roots are found in case of monocots. All right, so you can say that roots originating from base of stem is called fibrous root. So that is called as fibrous root, and these fibrous roots are found in monocots. Monocotyledonous plants of the angiosperms. All right. So this is all about the questions that we have seen and we have discussed it. Thank you.